Hello, and welcome back to another episode of reading just any old science article that is interesting to me. And this one here is a recent one from, I guess it was from published online in October. And it is about adaptive radiation. And so, a continuous fish fossil record reveals key insights into adaptive radiation. And adaptive radiation is when one species rapidly diversifies and becomes a bunch of different other species. And so, I guess hopefully through this article, we can learn, some of the mechanics of evolution and how evolution could take place. And that's such a interesting subject to me. So we're just going to get right into it. Adaptive radiations are a major component of biological diversity, and their subject provides insight into the structure and evolutionary dynamics of biodiversity. Adaptive radiation is the diversification of a lineage into an array of species that possess traits enabling them to exploit different environments and resources. It involves a complex set of processes, and identifying the factors intrinsic and extrinsic and intrinsic that initiate, facilitate, and constrain them remains challenging because of the difficulty of reconstructing conditions at the onset of classical large radiations. Comparative studies of adaptive radiations provide evidence that ecological opportunity is key to diversification. So I guess that would be a ecological opportunity. An example of that would be like when a fish realizes it can go on land. Ecological opportunity refers to the opening up of new niches and the new availability of an abundance of resources. It can trigger diversification through ecological release and ecological expansion, subsequently followed by specialization with niche partitioning during or soon after speciation. Ecological opportunity may lead to a burst of ecological diversification and speciation events, and may be triggered in either several ways, including 1. The colonization of a newly formed environment. 2. The emergence of new resources. 3. The extinction of predators or competitors. And 4. The evolution of a key innovation. All of these factors open up for resources that were previously inaccessible to diversifying lineage. So just with a fish being able to walk and breathe on land, I think that checks all these boxes. It's a new environment. They're finding new food. They're removing themselves from predators, and the evolution of key innovation. Maybe not that one, but that probably came later. Or I guess the legs would count as that. The ability of colonizing species to be able to access new resources depends on a species' ecological versatility and timing of arrival relative to other species. Ecologically, more versatile taxa can populate a wider range of habitats more quickly and can often access a wider range of dietary resources in individual habitats, which result in an increased abundance and wider distribution ranges. More versatile taxa. Wonder what they mean by that. If that just means like animals that are more resourceful. Ecological versatility. Ecological versatility is a prerequisite for rapidly accessing new resources when they first emerge, and thus versatile taxa may experience more immediate opportunities to diversify. The other often invoked but rarely tested hypothesis is that the arrival order of species can determine which one goes on to radiate. This is known as the priority effect, which holds on to early colonizing taxa can fully access, which holds that early colonizing taxa can fully access and subsequently monopolize resources, thereby negatively affecting the opportunities for taxa that arrive later. For instance, it has been shown in microbial diversification experiments that early arriving species suppress the diversification of late arriving species. However, testing for priority effects and/or differential versatility of colonizing lineages in real adaptive radiations has proved elusive because many of the best studied adaptive radiations, including most cichlid fish radiations, began millions of years ago, which makes it almost impossible to access a rival order or differential, which makes it almost impossible to assess a rival order or differential ancestral versatility. Nice. They're still here. Lake Victoria cichlid fish. I gotta look up this pronunciation.
Cichlid.
The Lake Victoria cichlid fish radiation provides us with unique opportunities in this regard because it unfolded over an exceptionally short and recent geological time interval. Genomic analysis of Lake Victoria radiation show that much of its genetic diversity stems from hybridization events between two lineages, which had been evolving separately for millions of years. Although this event predates the formation of modern Lake Victoria at approximately 17,000 years ago, the genetic variation was present in the founding haplochromine lineage. What is that? Haplochromine lineage. The haplochromine cichlids are a tribe of cichlids subfamily. These fish look really cool. Beautiful. As this lineage diversified, it left a trail of accessible fossils in its wake. The sedimentary record of Lake Victoria provides excellent chronological resolution from which we have attained rich and continuous fossil record for this study. Previous studies used phylogenetic reconstructions and attributed the rapid evolution of large species diversity in the haplochromine cichlids to ecological opportunity, the emergence of large deep lakes, a highly evolvable mating trait system, and unusual genomic potential due to hybrid ancestry. So what do they mean by a highly evolvable mating trait system? And this hybrid ancestry, that's interesting. But I wanted to look at what a phylogenetic reconstruction was. A phylogeny is the evolutionary history of a group of entities. Given that this can only be known in exceptional circumstances, the main aim of phylogeny is to describe evolutionary relationships. Okay. However, comparative phylogenetic approaches do not inform us about past community composition if of taxa other than the target clade, and therefore about relative versatility and arrival order. But such data can be obtained in principle from fossils and their abundances and distributions over habitats and through time. Here we reconstruct the history of arrival order and the dynamic history of the relative abundances of radiating taxon and all other major fish taxa across habitats from the origins of the lake to the present. We took a series of 4 long composite sediment cores along an onshore to offshore transect of the eastern part of Lake Victoria. First, we asked whether the radiating lineage arrived in the lake before other taxa and whether or not it was numerically dominant over the others. Both early and numeric dominance could afford haplochromine cichlids the opportunity to monopolize resources, permitting them to radiate while potentially inhibiting the radiation from other taxa. Second, we test whether the ancestors of the radiation differed from other contemporaneous taxa in relation to their early habitat utilization, specifically their ability to persist in new lacustrine habitats that emerged as lake levels rose that have no analog in small streams and swamps from which all colonizing fishes came. Okay, I was wondering how these fish got to the lake in the first place. So they came from streams and swamps, and then these lakes rose. Lacustrine is a word I don't know. Oh, permanently flooded lakes and reservoirs. And they have Lake Tahoe as an example. Cool. Based on more than 7,500 fish tooth fossils identified to either family level and or major lineage within the cichlid family, our evidence suggests that both radiating and non-radiating lineages co-colonized as soon as the lake began to form, and the radiating taxon did not dominate the assemblage. However, when the lake became deep and when vast open water habitats formed, haplochromine cichlids established in offshore deep waters and instantaneously dominated the fish assemblage in these new habitats. In contrast, the hitherto-numerically dominant crypanoids and all other taxa tracked the littoral zone but successfully abandoned each site as it soon transitioned to deep water pelagic habitat. Wow. Okay, this is a lot of crypanoids. That sounds like an alien. Fish includes carps. Okay. So, some big fish. Nice. Littoral zone. The shallow downsloping shelf of a lake or pond. So they're finding that so these fossils, even as the lakes expand, the carps or their ancestors had to stay close to the littoral zone, the shallow edge of the lake. And this word too, pelagic habitat. Pelagic habitats create the foundation for marine food webs, home to primary producers such as phytoplankton and microbes. Nice. This suggests that cichlids, and in particular haplochromide cichlids, differed from all other fish that colonized Lake Victoria in their exceptionally large ancestral ecological versatility in relation to habitat. We suggest that the relaxation of selection on traits associated with habitat utilization, that is, ecological opportunity, and the subsequent population expansion into new deep and open water habitats were crucial for the rapid adaptive radiation of these fishes. Priority effects associated with the order of arrival and the relative numerical dominance at the origin of the lake cannot explain which lineages radiated and which did not. In the following, we derive these conclusions in more detail. A continuous fish fossil record. We recovered 7,623 fish tooth fossils from our different coring sites, together spanning the 17,000 acre refilling history of Lake Victoria after the late Pleistocene desiccation. Late Pleistocene desiccation. That's a good one to know. A total of 931,000. Whoa, oh my gosh. Okay, that's not one number. That makes sense. 931, 1,368, and 2,842, and 2,478 fossil teeth were recovered from sieved samples from the coring sites, and here are the names of the 4 coring sites: LV4, LV1, LV2, and LV3. From the deepest, the fossil teeth recovered were generally small, often less than 1 millimeter in length, but were usually very well preserved. Teeth were present in good numbers throughout each of the sediment cores, which permitted the continuous reconstruction of fish communities in each site. Although other fish remains, such as bones and scales, were found too, we present only the fish tooth fossil results here, as nearly all teeth can be morphologically assigned to a taxon with certainty, something that is not possible with bones or scales. Yeah, that is so crazy that they can just someone who's a professional in this field can just literally take one tooth and identify the animal that is. That just absolutely blows my mind. Generally, we observed high fossil abundance in periods of time where the site was a productive wetland or littoral zone, followed by a drop in numbers when the same site became a deep open lake habitat. This expected productivity gradient through time repeats itself as a gradient in space across the 4 cores, where the cumulative abundance increases from the offshore to the inshore. Makes sense? It's easier to live where the water is warmer, more plants, things that eat the plants, more prey. That makes sense. The fish community assembly. The diagnostic characteristics of tooth fossils were indicated that we recovered representatives of 5 fish families. I'm going to butcher these names. The Bagridae, the Claridae, the Mocochidae, the Cyprinoidae, and the Cyclidae. with haplochromine and aureochromine cichlids diagnosably distinct, which matched the dominant elements of the modern fish community in the lake. A small proportion of fish teeth could not be assigned to any taxon. Bagrid, clarid, mokochid catfish made up to 0.12, 0.13, and 0.24% of the total number of fish of fossil teeth, respectively. The overall percentage of the cryptonoid fossils in the assemblage was 4.53% across all 4 sites, whereas the entire cichlid family comprised greater than 80% of the assemblage of the site across all 4 sites. The haplochromine cichlids contributed 82%, and the aureochromine cichlids 4.9%. This 4.9%. This overall composition of the 6 lineage closely resembles the relative abundance of taxa in contemporary Lake Victoria. However, relative abundances were very different in the first few thousand years of the lake's history. The continuous fossil record demonstrates the haplochromine cichlid fishes, albeit always present in the sites throughout the lake's history, did not come to dominate the assemblage until several thousand years into its history. In fact, the cyprinoid fish dominated when the lake began to fill. For several thousand years afterwards, all other taxa were also present at the origin of the lake but declined later, often below the detection threshold. So let's look at figure 1. Lake Victoria fish assemblages since the birth of the modern lake, reconstructed from sedimentary deposition of tooth fossils. Calendar years. So here's the haplochromie. This is their boy that they're really looking at. Oh, he's over here too. Okay. So estimated fish. Okay, these are the different core sites. And yeah, what's interesting about, it seems like they're pretty constant throughout. But the cryptonoidae just always drop off, it seems like. Huh. But then they also have a rise here, so I wonder what that could be. So this is the so green is the wetland fish community. That's interesting. So for the first few thousand years, they're saying that the cryptonoids, sorry, I'm not saying that right. The carps dominated for the first few thousand years still, but eventually, I don't know, I wonder what happened here. This is kind of hard to read. All right. Habitat transitions and community turnover. The Lake Victoria Basin is estimated to be approximately 400,000 years old, with a history of drying and refilling, with the last event of complete desiccation in the last glacial maximum approximately I need to learn how to read this. This Ka, I thought that was this might be years. No. Ka time History. No. Paleontology. This is a disaster. And it started re-wetting approximately 17 Ka. The deepest point of the lake is 68 meters, and our deepest core, LV, is 63 meters. Our deepest core reveals subaerial exposed deposits of Paleovertisols, possibly with local episodic wetlands, which suggest very dry conditions from at least 20 Ka ending in 16 Ka at the latest. At about 16 Ka, shallow water and wetland conditions established, characterized by abundant typha pollen, which suggests the in situ typha growth. This condition lasted for approximately 2,000 years until about 14,000 BP. The topography of Lake Victoria is well characterized and is essentially saucer shaped. From when LV4 began to be inundated, we found continuous traces of fish fossils from sediment cores, and this allowed us to identify major shifts through time in the taxon composition of Lake Victoria fish community, statistically supported by change point detection analysis. We identified several distinct phases between the change points and assigned major habitat and ecosystem stages. Within each stage, we compared the abundance of fish taxon between these stages and inferred the direction of change between the stages using t-tests. To be able to convert fossil tooth counts into relative abundances of fish of different taxa, we accessed published information and our own computerized tomography scans from preserved fish to obtain information on the number of teeth that typical representatives of each major taxon have. Tomography, what is that? Tomography.
Tomography.
A technique displaying a representation of a cross-section through the human body or other solid objects using ultrasound. Sweet. Four major fish habitat zones are recognized in modern Lake Victoria: wetlands, littoral, sublittoral, and deep water. Whoa. Palynological and paleolimnological data. Jeez. Just throwing out these words. Study of pollen. Oh, that is really cool. I love pictures like these. Limnological. What would that be? Something to do with rocks, I think. Sounds familiar. Focuses on reconstructing past environments of inland waters. Okay. The first site to be inundated with the onset of the modern lake suggests that the site was situated in a wetland with low water levels for approximately 16.6 Ka until approximately 13.5 Ka. An abundance of pollen of semi-aquatic macrophytes and hydrophytes suggest a wetland interspersed with shallow ponds and low water tables until approximately 13.5 Ka. 6 fish groups found in Figure 1 are found in Figure 1. Cyprinoidea was clearly the most abundant group, followed by haplochromine and aureochromine cichlids, and finally mochiochid, bragid, and clarid catfishes. This composition resembles that of modern wetlands and shallow lakes across much of eastern and central Africa. We refer to this wetland stage as defined by the fish community as Phase A. By approximately 13 Ka, typha pollen disappeared and rainforest trees began to spread, which suggests an increase in the precipitation/evaporation ratio, and Site 4 began now being distant from the wetlands because of a corresponding change in pollen records, with the disappearance of typha was observed simultaneously in Site LV2, which is 40 meters shallower than LV4. We interpreted it as a signal of our coring sites transitioning to offshore habitat in a deeper lake. So I think what they're saying here is that they can tell they're moving into deeper lake territory because of the absence of the pollen that they were seeing in previous samples. A recent attempt at reconstructing lake levels suggested that LV4 became a deep open lake habitat just before the change in pollen composition. Around the point in time when pollen records suggest a transition from a wetland to an offshore lake site for LV4, many fish taxa declined to somewhat lower abundance in the sediment record, including the previously numerous cyprinoids. This change is statistically corroborated by the detection of a first multivariate change point at approximately 13.5 Ka, associated with a significant decrease in cyprinoids, but not haplochromine cichlids. Subsequently, the catfish disappear from the record and the community is composed only of cichlids and cyprinoids. We interpret this community as reflecting a period of lacustrine littoral conditions at the site, which we refer to as Phase B. It lasts from approximately 13.5 Ka to approximately 11.9 Ka. When we detect another significant change point, at 11.9 Ka, they detect another significant change point. At this point in time, most fish taxa declined to below detection level in the sediment record, including the previous numerous cyprinoids. However, while cyprinoids in all taxa other than the haplochromines fell below detection level threshold, we consistently recovered haplochromine cichlid teeth from sediments younger than 11.9 Ka. This community then resembled the modern community in offshore Lake Victoria, except that it lacks evidence of pelagic cyprinoid Rastronobola that is abundant in the modern Lake Victoria. Oh, these fish seem very small. Maybe that's not right. We refer to this deep open lake community as Phase C community, defined by the dominance of the haplochromines and a nearly complete absence of cyprinoids and catfish. At the base of our second deepest core, LV1, at approximately 13.8 Ka, we detected a short interval with a community that resembled the wetland community at the base of LV4, with cyprinoids and haplochromines, and the presence of the macocid, bagrid, clarid catfish, and oreochromid cichlids, whereas the cyprinoids had numerically dominated between 16.6 and 13.5 Ka. Cyprinoids and haplochromines occur in smaller numbers at the base of LV1, resembling phase B in LV4. Hence, site LV1 appears to have been a productive wetland for its first 1 Ka, and after which its fish community suggests that the site transitioned to a lake. Just as in LV4, after approximately 13 Ka, all taxa abruptly declined below detection limit except the haplochromine cichlids, which declined but remained above the detection limit. This change is reflected by the detection of a multivariate change point at approximately 11.8 Ka, which, however, is placed approximately 1 Ka after the visually detectable community change. For this reason, we assign the section below the change point to a heterogeneous phase, which the wetland transitions to a shallow lake littoral. Our data from core LV2 tells a similar story. This core starts approximately at 15.5 Ka, and the first 3,000 years of the characteristics of wetland communities described above: codominance of cyprinoids and haplochromines, and an abundance of oreochromines, as well as regular presence of various catfish. This community persisted from approximately 15.5 to 12.6 Ka, which is where we detected a multivariant change point associated with declining abundances of all taxa. Subsequently, the catfish disappear from the record, and the community is composed of only cichlids and cyprinoids. We interpret this community as reflecting a period of lacustrine littoral conditions at the site, that is, phase B. It lasts approximately 12.6 Ka to approximately 10.4 Ka, where we detect another significant change point. After this, this site LV2, like LV4 and LV1, loses all fish except for the haplochromines and occasional oreochromine cichlids. We interpret this as the transition to the deep pelagic lake condition, and it is marked by a significant change point around approximately 10.4 Ka. This coincides closely with an abrupt drop in the hydrophyte pollen, an increase in rainforest tree pollen, and an abrupt increase in the ratio of rainforest to savanna pollen, albeit less steep. Yeah, the fact that pollen just stays in its form for the millions and millions of years that we need to look at it, that we need for us to be able to look at it, is amazing. As it just tells us all the different plants that were growing around there. Finally, our record for LV1, our coring site closest to the shore, starts at approximately 11.5 Ka, and must have been situated within the littoral and/or sublittoral zone of Lake Victoria. Though throughout its history, consistent with this, we found fossil teeth of haplochromine cichlids, aureochromine cichlids, cyprinoids, and cyprinoids deposited in the sediment throughout the record. We observed dynamic changes in the abundance of all these taxa as reflected by several significant multivariate change points. The dynamic changes are also reflected in significant t-tests. We conclude that this site has not had any lasting wetland phase, but became first incorporated into an expanding lake as a littoral zone and remained littoral or sublittoral throughout the lake's history. Most notably, the abundances of cichlids and cyprinoids increase around 6 Ka BP. Here's BP. Six Ka BP right here. I think they're talking about this. That's cool. So here it looks like these are all the fish teeth, which is so crazy. The cichlid's ecological versatility. Between 13 and 12 Ka, the profoundal and the pelagic zones, deep and open water habitats, emerged as totally new habitats that had no analog in the old stream and wetland landscape that would have constituted fish habitat in the preceding millennia. These new habitats would have potentially provided the ecological opportunity for all fish taxa inhabiting the lake at the time. However, the abundance of all fish except cichlids dropped below the detection thresholds as the site transitioned from a wetland and or shallow lake into the new habitat. So why did the cichlids succeed and the other fish fail? This suggests that although various species of fish successfully established in shallow water habitats when the modern lake began to fill, of all the fish in the ecosystem, only cichlids possessed the ecological versatility that allowed them to persist in the new deep and offshore environments. This is consistent with the structure of modern Lake Victoria fish community, where most groups are confined to the nearshore littoral and sublittoral zones, whereas haplochromines occur in large numbers, and there may be many different species in all habitats across a full depth range that compare to the complete and the this is consistent with the structure of modern Lake Victoria fish communities, where most groups are confined to the nearshore littoral and sublittoral zones, whereas haplochromines occur in large numbers and there are many different species in all habitats across the full depth range and the complete onshore-offshore gradient. It is also consistent with data that show the riverine haplochromines recently introduced into a deep crater lake abound there in the deep offshore habitats. Our fossil evidence suggests that the intermediate prevalence of haplochromines in the new environments as they emerge cannot be a result of evolutionary adaptation during the adaptive radiation process. If this were the case, we would have expected a period of absence of haplochromines followed by their return to the new habitat. The uninterrupted presence of haplochromines suggests that their ability to persist in the new habitat was already present at the beginning. Clearly, haplochromines, and to a lesser extent aureochromines as well, arrived with large habitat versatility that none of the other taxa possessed. Cichlid fishes have been suggested to exhibit unusual ecological versatility with respect to their trophic ecology. trophic. I guess it's just saying what they eat. Study of the feeding relationships. Okay. Their respect to their trophic ecology, habitat utilization, and associated breeding ecology, and have highly versatile feeding apparatus. They have been shown to occupy a very wide range of freshwater habitats from little streams and swamps to large and deep lakes, and a single species can occupy an entire depth range. Our fossil data suggests that the haplochromine and oreochromine cichlids persisted in all of our offshore sites when the sites became deep offshore lake habitats, whereas other fish groups disappeared despite having been numerous at the same sites when they were shallow habitats. We consider this as evidence of the differential habitat versatility. Although the oreochromine cichlids displayed ecological versatility too, their numbers decreased to a near detection limit in our two most offshore sites, which is consistent with the modern ecology of the two native species that mainly occurred in the near shore parts of the lake before their expatriation. Extirpation. Water depth and onshore offshore habitat gradients play a fundamental role in haplochromine speciation. Therefore, it is likely that their large ancestral habitat versatility was key to the rapid emergence of adaptive radiation because it exposed haplochromine populations very quickly to large ecological constraints to which they would subsequently adapt. The fact that only haplochromines radiated into approximately 500 endemic species, but oreochromines did not, suggests that although required versatility alone was likely to be insufficient for adaptive radiation, it is possible that its coincidence with large evolvability due to hybrid ancestry. Due to the hybrid ancestry of the haplochromine lineage, was key for converting a single ecologically versatile population into many species with distinct ecological speciations. Ancient hybridization fuels rapid cichlid fish adapt. What is ancient hybridization? What is that? That's crazy. Hybrid ancestry. What does that even mean? That's interesting. So it's kind of just means what it means. Hybrid obviously is it's two new species being created by two separate species. In all of the late emergence of pelagic abundance, in all of our offshore cores, we observed a late Holocene increase in the abundance of haplochromine cichlids, followed by a partial return of the cyprinoids to offshore waters. The multivariate change point analysis places this change in all sites between approximately 3.8 and 2.3 Ka. But visual inspection of the data is consistent with quasi-simultaneous change in all three cores around 3.5 to 3 Ka. A significant increase of haplochromines in all three cores was confirmed by t-tests, and that's just a probability test. We refer to this late Holocene pelagic abundance phase as phase D. Intriguingly, demographic analysis of the whole genome sequence data from modern samples of all haplochromine trophic groups suggests that specialized pelagic planktivores species that eat plankton, emerged only in late Holocene, which would coincide with a significant increase in haplochromine cichlids in our offshore core sites. Palynological reconstructions revealed that approximately 5 Ka, the riparian vegetation changed from rainforest back to savanna with a significant increase in charcoal influx in all three cores. Where would the charcoal come from? These changes in terrestrial vegetation cover mark the end of the African humid period, reflect long-term changes in rainfall and wind patterns, and may have led to increased productivity in the lake. It is noteworthy that shortly after the cichlids increase in offshore cores, cyprinoids reappear in the record after they had been below the detection threshold for several thousand years. Modern Lake Victoria has one pelagic zooplanktivorous. What the fuck? What the heck? Zooplanktivorous. Zooplankton. Okay. Animal that eats plankton. This is the only species. It says modern Lake Victoria just has this species in there that eats plankton. The silver cyprinid. Wow, it's cool. We hypothesize that our fossil record that our fossils represent this species and that its pelagic habitat may have emerged around this time, which is something we hope to address with DNA in the future. All right, conclusions. Our study unravels the interaction and relative importance of key facilitators of adaptive radiation at the onset of large adaptive radiation. Since haplochromines diversified into hundreds of endemic species in very little time, whereas none of the other lineages did, one might have expected haplochromines to have arrived before others or to have acquired dominance through abundance before others. Our evidence suggests that neither of these were the case. We show that when the most offshore coring location became inundated, the fish community immediately resembled modern assemblages of wetlands and rivers in the region in astonishing detail. With an abundance of cyprinoids, haplochromine and aureochromine cichlids, and several catfish taxa. This may seem surprising at first, but it is not so on closer inspection. Large lakes with endemic species radiation are often linked to offshore islands in the sea. However, contrary to the latter, most large lakes do not start out as geographically isolated from the source region from which they from where they get colonized. In fact, large lakes form in the midst of the mainland, which is the river network large lakes form in the midst of the mainland, which is the river network in the case of freshwater fish. Hence, we should expect many different river fish lineages to colonize newly forming lakes synchronously. This also means that there's little opportunity for priority effects to determine which colonist's lineage radiates, and this factor should play less of a role than for radiations in ocean islands. In turn, lineage-specific effects should be more important to explain which lineage radiates in the lakes. The duration of wetland fish assemblage in Lake Victoria's history closely coincides with a dominance of wetland and savanna vegetation around the lake, based on pollen records, and it is lost as soon as the vegetation indicates the emergence of a deep lake. The ability of the haplochromine cichlids to fully occupy the new habitats associated with the development of deep lacustrine conditions set them apart from all other colonizing fish. We propose that this ecological versatility in the new deep ocean open water habitat has played a decisive role in the diversification process and led to the subsequent ecological dominance of haplochromines. We suggest that the coincidence of ecological opportunity with ecological versatility combined with a hybrid ancestry was paramount to setting the stage for the massive and explosive adaptive radiation that occurred in Lake Victoria. Without both opportunity and versatility, the hybrid ancestry of the lineage would have been unlikely to lead to massive diversification. Opportunity and versatility in turn would have been unlikely to convert the versatile population into an exceptional radiation, which required large evolvability and hence large genetic variation. Our findings add to understanding the interactions between environmental and lineage-specific factors at the onset of animal adaptive radiations. We suggest that adaptive radiations may be triggered by the coincidence of ecological opportunity with the intrinsic lineage traits of versatility and evolvability. And that is this study here. Let's see if they have any more pictures anywhere. Doesn't look like it. Okay. So in this study, basically what they're saying is there were these rivers in Africa, and the cyprinoids were a group of fish that really dominated these rivers. And then as Lake Victoria started to form, as these deeper lakes started to form, it ended up being the haplochromines that were able to dominate in these new ecosystems. And the conventional wisdom of ecology, what they're saying, the study is saying the conventional wisdom of ecology would have thought that the first fish to enter the new habitat would be the one that dominates. But they believe from their fossil record and just from thinking about how it would happen that all these different fish species from the rivers are entering these lakes at the same time. So they don't think that the haplochromines were able to dominate like immediately, but for some reason, and they said they're going to explore that in the future. The haplochromines were able to kind of do a coup in terms of abundance to the cyprinoids, and they mentioned their hybrid ancestry, which sounds really interesting to me. I don't know if just having like a more diverse genetics in the fish just makes them better, makes them more able to adapt to the new environment. But yeah, no, I thought this article is pretty interesting. And if you did